In this monograph on Müller cells, you have heard the excellent introductory presentation by Professor Leuenberger and the three beautiful lectures by Stella Vujosevic, Feliciana Mena, and Maria Cristina Savastano. You are now much more knowledgeable on this subject at this point, and in order to be able to delve further into the subject, we need to consider that Müller cells are, however, part of the glia, and without at least a cursory knowledge of the cerebral glia from which the retina descends, it is impossible to speak the same language. I therefore asked Professor Gabriella Biella, an eminent connoisseur of the subject, and with whom I shared the same violin professor for 10 years, our beloved Maria Cristina André, to draw up an excursus that would not only serve as a spelling book, but would also allow us to build new bridges in research in this field. A luxurious, special, tailored presentation for ophthalmology residents, an unmissable excursus for neurology residents. Here the positioning of its presentation in the context of the work. Professor Biella is experimental neurophysiologist and neuropathologist, former head of research at the Consiglio Nazionale delle Ricerche, member of the BTPA panel committee at the European Synchrotron Radiation Facility, Grenoble, France, visiting scientist at the ESRF, visiting scientist at the Paul Scherrer Institute in Willigen here in Switzerland, in the neighbor of Zurich, and is therefore extremely qualified to talk to us on this subject. Good listening. Here are the contents.
This talk will regard the so-called macroglial components of the brain. With this term, I will indicate the two major populations that compose the glial populations of the central nervous system, and namely the oligodendrocytes and the astrocytes. This talk is a bit an out-of-the-soul lecture in the context of the lectures and lessons on Müller retinal glial cells, that is the marker of your group of study just on this kind of special glial retinal cells. And due to the fact that the literature on oligodendrocytes and astrocytes in the brain is definitely an enormous amount, composed by an enormous amount of works, this will serve as a suggestion for future studies on Müller retinal glial cells and for your group next studies and study addresses. In this picture is represented the cover of the scientific and medical journal Wissenschaftliche Medizin, edited in Frankfurt am Main in 1856. And in this issue is reported the essay by Rudolf Virchow on the so-called Nervenkit, what he called the Nervenkit. That means the glue, or it comes just directly from the ancient Greek gluon, that is the sticky matter maintaining the structure and the organization of the neural components of the nervous system. And this name of Nervenkit has been then transformed in glia, just in glia, just for the sticky appearance of the central nervous system, both in humans and in mammals, and not only, when extracted from the skull. And after the first observations by Virchow, there have been further studies who have been impressively important to define the two major populations of glia, that is made, as we said before, by astrocytes and oligodendrocytes. These two classes of glial cells, together with microglia, represent globally what is now called the glia or glia. It depends on the region in the Anglo-Saxon world when you read this word. And the microglia represents a main immunoresponsive component of the macroglial elements, and they are specific cells that we will describe in a brief presentation at the end of this talk. The macroglia is composed so by astrocytes and by oligodendrocytes, and the third component that we'll treat but in few images is the microglia. One first problem about these kinds of populations is that in the brain, gray and white matter, GM and WM respectively, we can find a significantly different composition in terms of these two major populations, where sometimes oligodendrocytes are much more represented, and in other parts of the brain, astrocytes are the major components. I will give here a very general introductory and sparse note list. This is meant as a purely introductory and as a scattered concept introduction to fundamental concepts to be used in deeper analysis on glial cells during this book. Oligodendrocytes and astrocytes, as we said, are differently distributed in the central nervous system. Just an example. If we take into consideration the human frontal cortex, we have there, as we all know, a white matter part and a gray matter part of the cortex itself. And it is interesting to note, this is a pure example, but we can extend this example to the whole brain. Oligodendrocytes are more represented in the human frontal white matter, while astrocytes are a diffuse minority. Why? But astrocytes definitely outnumber oligodendrocytes in the gray matter of the human frontal cortex. So we have a problem, not a problem, but a question open on glial diffusion asymmetries. And notwithstanding, we can understand, and we will understand why these asymmetries are so common. Many problems opened by these questions are still under scrutiny and are a suggestive problem for a rich array of interpretation in future studies. Here we can skip immediately to a general developmental issue list for neurons and glial cells. And as you can see from the picture, we have a very strange picture with a curved line on the top, signed by the term leptomeninges, and a basal straight line where there is the description of ventricle. And the abscissa lines, let us call them, even they are not strictly abscissae, even if they are strictly related to the time of development of the brain, are representing the neurogenesis and the gliogenesis stages. As we can see on the very left of this figure, the short distance between the leptomeninges and the ventricle surface is a small distance. This is because the brain is beginning to evolve and to enlarge, and the enlarging steps of the brain are represented by the curved, upsurging black line that is represented by the leptomeninge, indicator. A second interesting and important issue is that the basal straight line indicates the ventricle. The ventricle in the brain represents a kind of region where all the brain cells take origin during the development. And the third interesting issue is that we have the neurogenesis stages preceding the gliogenesis stages that happens in the late part of the CNS, the central nervous system development. And now let's have some more detailed indication of all the acronyms constellating this figure. The first important thing to bear in mind is that, as it is written on the top of the written part of this figure, is that glial cells stem from a neuroglial common progenitor. What does it mean? That we have a common progenitor for glial cells and neurons. That means that neurons and glial cells have really a common stemming line of cells. And then only in further steps they divide and they become what their fate is written in their genetic patrimony. And as we can see here, the common neuroglia progenitor cells are signed by this red circle in the figure on your extreme left, and the acronym is NEC. That means just neuroepithelial cells. These cells, as you can see easily from the text, undergo symmetric cell divisions to expand this initial pool that is not so rich, but it expands very, very rapidly and with a very potent dividing power. And they expand this initial pool and later transitate into a second stage that is represented by the blue circle around the VRG S or C cells, that mean the ventricular radial glial cells. This is the first important stage that these VRGCs that are signed in red throughout the whole picture represent a kind of bridge between the ventricle and surface and the leptomeninges. And these bridges will serve as rails or scaffolds where the nascent progenitor of neurons that are signed here either in green or in blue are climbing upon these lines created by these special astrocytes that are connecting the two sides of the basal and the apical side of the developing brain. And the asymmetric cell division go and generate a second VRGC generation that is a second generation of ventricular radial glial cells that allow for this climbing from the ventricular zone, as it is signed here, up to the cortical plate as indicated by the greenish circle in the figure. And these kind of bridges are the first true astrocytes of the brain, but they are not a stable population, because stable populations of astrocytes will develop and mature in later stages, as we will see in the next presentation, the next step of this talk. And as you can see, so we have this kind of astrocytes, the glial cells, the original glial cells that connect the two sides, from the depth to the top, the ventricle to the leptomeninges. The early born projection neurons, the PNS, that climb along this kind of glial cells, that are this kind of astrocytic progenitor cells and astrocytic radial cells. These early born projection neurons, or PNS, that are represented by the acronym PN here, go and inhabit the deep layers of the cortex that are signed here in the rounded rectangle enclosing the L5 and L6 layers of the cortical layers. I wish to underline, but it is very evident to all of you, that the symbols from L1 to L6 are the 6 layers of the maturing cortex. It is interesting to note that the deeper layers of the cortex are the most precocious populated regions by neurons in the brain, in the cortex, in comparison to the superficial layers that are represented by later born neurons that are directed to the upper layers of the cortex itself, and these are signed by this rectangle enclosing the 4 layers from L1 to L4. Additionally, some populations of these radial glial cell daughter cells convert themselves into intermediate progenitor cells, that is the IPC, signed here by this amaranth circle around the IPC acronym and the red cells. We can see throughout the figure other IPC cells that comes from the original radial glial cells in orange, just almost in the second third of the figure. And these intermediate progenitor cells represent and go to form then the outer radial glial cells, or ORG, that is represented here by the black circle. And they go also in the subventricular zone, the SVZ, as here, and only in the latest stages of development of this very complicated migration scheme of cells composed by neurons, astrocytes, and oligodendrocytes, there is the gliogenesis stage that is represented here by the round-shaped rectangle enclosing the word gliogenesis. In fact, after the neurogenic stages, gliogenesis occurs, generating then astrocytes and oligodendrocytes, as it is very evident from the figure. Let's get more in depth on this gliogenesis stages. Let's have a fast repetition of the figure illustrated in the previous slide, showing the radial glial progenitor cells, also called radial glia. Radial means, as many of you will have well understood, the kind of radium going from the reddish and rose cells representing the radial glial cells, getting up to the pile surface with mature interneurons and some other neurons already climbed from the radial glial cells up to the cortical or subcortical targets they are devoted to. And one important thing is that the body of these spatial astrocytes that are the radial glial progenitor cells reside in the embryonic ventricular zone, as we said. But we have a much clearer image showing the neonatal subventricular zone or ventricular zone, and all around there are the neural stem cells and the radial glial cells, and even the neuroblasts that begin their ascent to the surface or to the mid-surface layers of the brain. And as we said, during development, the newborn neurons use just radial glial cells as a scaffold to reach their final destination. Here in the orange rounded rectangle are enclosed a number of these nascent neurons, neuroblasts, climbing and getting their destination. And it is a very fast because we must come here to a more important figure, and we have the image on your left representing in a less descriptive way, but in a conceptual way, the interrelationships between the glial cells and the neuronal cells. The glial cells in the central part of the figure are represented by the green cells or the green ovals on the bottom of the central figure. And in gray are represented the neurons. And the figure is in some way ideally represented the four stages that are composing the cortex during these stages. And there is at the bottom the ventricular zone, then the subventricular zone, SVZ, then with IZ is indicated the intermediate zone, and finally the cortical plate. The central figure has to be analyzed more in detail, and these details are well represented by the three rectangles imaged on the picture. There is a top rectangle and two lower, the left and right rectangles, indicating the crucial regions of this dynamic picture. And there are several acronyms indicating, in this case, the genes that are involved in the development of this extremely complex process. And as we can see at the higher magnified, in the higher magnified rectangles, upper, lower left, and lower right, I have shown here three lines, the red line, the maroon line, and the light azure line, indicating 1, 2, and 3, where there are in some way in detail indicated the gene families that are involved in the orchestrating this impressively complex process of the brain development. And as you can see in the written text, there is in the pile surface written in Amaranth, the families of genes participating to the attachment of the terminals of the radial glia, of the radial glial arrangements to the pile to the leptomeninges surface, and then little by little, I don't stay here to describe them in details, but they are easily readable in the text and in the figure. We have genes devoted to base local adherence junctions in black in the right bottom enlargement figures of magnified rectangle, and in the third rectangle we have a mitosis that represents a kind of division of the original astroglial cells, and in the black column in the midst of this third figure is representing the chromosomes accumulating in the midst of the mitotic fuse of the mitotic spindle, and they are proximate to the stage of division in two populations to generate the two new astrocytic cells. And we are observing it in this scheme as it were in the metaphase plate reported in black. Just because it is a extremely complex picture. I show here a very simple summary of the genes involved in the development of this crucial dynamics of the brain, and especially of the cortical part of the brain. And as you can see easily, all the genes numbered and populating the three rectangles that represent the three crucial regions of this cortical development. And the first I tried to represent here are genes involved in radial glial cell basal processes and their attachment to the top pile surface that are represented in amaranth into the magnifying rectangle on the top of the figure. As you can see, it's a very complex family, and it has not to be considered a simple addition of gene interventions, but a configuration, a net of genes interventions. And the fact that the genes are intervening in interlaced actions, this means that the genes are a part of a very large net, and this makes the landscape of these events extremely complex. Then there are genes linked with other injunctions in black, and these are represented on the left lower rectangle with the sign like in mathematical equal. And these adherence junctions will be considered after to understand what they are and their functional meaning. There are genes participating in apical polarity attachment to the ventricular surface. We can see these kind of genes indicated in purple on the left lower rectangle, and they report that all the paper make is to call them apical polarity, and it is not an apex, it is a base of the cell, but the wrong denomination continues to remain in the literature. So let's call them again apical polarity, but we must be aware that we are on the basal structure of this complex, and they are indicated in purple. Then there are genes essentials for the primary cilia. Primary cilia are indicated in blue, and in the left lower rectangle are those blue little structures anchorating the complex of the apical polarity structure and the developing astrocytes to the ventricular surface. And the blue color gives a very good idea how these proteins allow for a hook anchorating on the ventricular surface or this complex. Then there are in the right bottom rectangles genes essential for the centrosome related structures. These are the two red circles, red little circles that are representing just the centrosomes that represent the terminal of the mitotic spindle that is indicated in light blue. And the mitotic spindle is a kind of rail system on which the chromosomes run during the mitosis. And we can see also that black structure within the right bottom rectangle, representing the dividing or divided chromosomes ready to go to their destiny, either on the left or on the right, to generate the new cells, the new astrocytic cells born from this mitosis. In green, and we must put our sight just on the greater figure on the center, there are represented genes that are involved in transcriptional regulation of the different astrocytic, or more correctly, astroblastic cells dividing and going to become radial astrocytes, and that these genes are WNT3A and PAX6. So there are many acronyms, many cells, many kinds of elements getting together all this complex picture. And one thing that I missed to quote are the cells reported in gray, that are the neuroblasts on the trails of the glial radial arms, or staying already in their position towards or within the cortical plate on the upper part of the central field. Just some sketchy notes on astrocytes and oligodendrocytes. Let's describe now some features representing astrocytes in gray and white matter areas of the central nervous system. The astrocytes are mainly of two types. There are protoplasmatic astrocytes, or P-astro, residing in the gray matter, and they are cells with a high degree of connections between them via gap junctional coupling. We will see after what this means. And they are mutually interconnected with other protoplasmatic astrocytes. This coupling is obtained by the intervention of many proteins, and particularly via two types of proteins called the CX43 and CX30. CX is the shorthand for connexin, and connexin 43 is also called gap junction alpha-1 protein, and is a protein that is a part of a gap junctions, intercellular communication among cells that regulate cell death, proliferation, and differentiation. And also connexin 30, that is devoted to other and parallel functions, has another name, that is gap junction beta-6 proteins. Then there are the fibrous astrocytes that are mainly present in the white matter, contrary to the previous type, the protoplasmatic astrocytes residing in the gray matter. The fibrous astrocytes have a limited coupling to other astrocytes and express only one type of protein for connecting to other cells, that is 6 or connexin 43. The connexin gene family codes for the protein subunits of gap junctions channels that mediate ions and metabolite diffusion between cytoplasm of adjacent cells. Let's now try to clear a bit this picture that has been only described by words. And in this figure we can observe the connexin, the hemichannel, and the gap junction structures. Connexin is a protein, and it has many proteins, not all proteins obviously. They can be host of the membranes around the cells, and passing the membrane and being proteins with a high degree of polarity, they must use specific tricks to be accepted by the membrane lipids signed in maroon in A. And we observe here a connexin protein with an N and a C terminal. And if we start from the letter N, we see a black curved segment that is the chain of the amino acids free in the intracellular compartment of the, in this case, of an astrocyte. And then in blue are signed all the intramembrane chains of amino acids pertaining to this protein. And as we can see, they are signed as cylinders. They are not actually cylinders, but the cylinder in some way tries to indicate the space that the amino acids in some way are able to express within the membrane in order to be accepted by the lipids of the membrane. And in fact, these blue segments are composed mainly by lipophilic amino acids. In this way, they can transit the membrane without any rejection. Then we have a blue line that is again an amino acid chain connecting to the other transmembrane domain of this connexin, and so on with an outside and inside chain getting through the membrane for 4 times, and we have the final amino acid chain terminating with the C atom. Well, this configuration is not the actual nature configuration within the membrane. We have here only 4 elements, but there are molecules made by many proteins, and there may be many transmembrane domains, more than 4, and usually 6 or even more. And we can see in B how these transmembrane domains are not separated in natural conditions, but they gather all together and to in some way isolate a central channel that is a free way from the outside to the inside and vice versa of the cellular membrane. And this connecting hexamer, in this case we have 6 transmembrane domains, we have the creation of a hemichannel. And the hemichannel is just the connexin protein, global polyprotein, identifying the molecule per se. But this is not enough, because we know that connexin, as we have seen, connexin 43 or 30, are able to allow for the junction of nearby astrocytes. This happens not only for astrocytes, but for many cells. In this case, we are considering astrocyte, and of astrocyte we will talk about. And we can see the 2 membranes. We can see cell membrane written on the extreme right of the figure. 2 membranes of 2 cells that can get in the nearbys thanks to the gathering of 2 hemichannels. And 2 hemichannels, one on one side in one cell membrane, and the other on the other side of the other cell membrane, gather together and unite to identify a double made hemichannel that is a full channel, that is called the gap junctional intercellular communication, or GJIC. As we saw before, connexin 43 can be called gap junction alpha 1 protein or gap junction beta 6 protein. And this gap junction have different names depending on the amino acidic components of the 2 protein clusters characterizing each hemichannel. And the 2 hemichannel identify a complete unique channel, and we will see how they work. They allow for outside to inside and vice versa transit of ions and metabolites. And we can see here in a magnified picture the gap junctions represented by the blue clusters in the figure, indicated by GJ, and this double clusters of gap junction. Each gap junction is formed by 2 hemichannels, and each hemichannel is made by connexin proteins, are allowing for the passage, for the transit in the opposite direction of soluble molecules, small molecules, and also by ions. As we can see, we can have a passive transit or an active transit with red and green molecules. And in the meantime, we can see at the extreme right of the figure in an oval red drawing the word stimuli. The stimuli can be of different origin, either electrical or chemical, and they induce a response in one of the 2 glial cells of the 2 astrocytes, and these responses can activate or inactivate many other metabolisms, and first of all the transit throughout the gap junctions from one cell to the other of the different substances needed in the task these astrocytes must do. In this schematic representation of oligodendrocyte types, we are now transiting from the astrocyte to oligodendrocytes. And we know that there are different types of oligodendrocytes in gray and in white matter areas of the central nervous system. The oligodendrocytes have a topographic differentiation in this way. Oligodendrocyte progenitor cells, OPCs, in GM and WM, display functional and metabolic differences. Just a few examples. Let us consider the figure in A, first of all, that we will see in much better detail in the next slide. In the white matter, more precursors and mature oligodendrocytes are present with slower turnover in white matter than in gray matter. A second difference shows or indicates that OPCs differentiate more efficiently in white matter than in gray matter. And finally, while precursor and full development oligodendrocytes in the gray matter display a higher number of glutamic acid receptors, AMPAs and CARs, we will see what they mean. OPCs in the white matter display a higher number of NMDA receptors. In addition, oligodendrocytes express day2 connexins. In this case, there are 632 and 647 other types of connexin that with 630-43 on astrocytes are particularly important for the developmental myelinization and oligodendrocyte survival in WM. Now, just because we presented many terms that are remained unexplained, let us see what they mean. Just in the A part of the figure, we can see an axon with the oligodendrocyte covering with myelin. And in the meantime, we can see the OPC, that means the precursor of the oligodendrocyte, taking contact with the nude axon that they are going to cover. And I ask you to observe in the extreme upper left of the figure the term aster, that means astrocytes, that remains in contact with the nude part of the fibers covered by two parts of myelin, two tunnels of myelin. And AMPAR function for CNS white matter glia in health and disease show these glutamate channels that are called just AMPAR, that mean alpha amino 3-hydroxy 5-methyl 4-isoxazole propionic acid receptor, acting as glutamate receptor. The illustration in A depicts the interaction of glial elements, as we said in CNS, white matter. Astrocytes and OPCs extend processes that make contact with unmyelinated axons and the nodes of Ranvier, the unmyelinated parts of the myelinated fibers. Now, don't panic, because all these issues will be discussed in great detail in the next parts of the talk. These glial processes contain functional glutamate receptors of the type AMPA. Cis white matter astrocytes exhibit AMPA-mediated CI2 calcium signals, and the precursors of oligodendrocytes display the AMPAR-mediated synaptic currents that are sodium currents regulated by the AMPAR receptor activated by glutamate. Glutamate releases that amyelinated axons drive synaptic inputs on the precursors, their OPCs. The functional consequences of astrocytes AMPARs in white matter remains unknown, but may include a role in the regulation of sodium currents. AMPAR activation influences many other functions. I remained here in surface because this slide otherwise would keep us for at least half an hour. AMPAR activation on OPC, in addition, influences the events in the nucleus of the cells, including the induction of immediate early genes. What does it mean? That on the precursors of oligodendrocytes, the activation by AMPAR receptors produces the true activation in the nucleus of these cells of these genes that activate with an early action and become the so-called immediate early genes. These are a very fast DNA response to an activation by external factors, and these are involved in cellular growth. Now let's consider this very simplified description of the figure indicated with B, where as we can see there are severe damages to the myelin covers and to the astrocytes and oligodendrocytes precursors. These are the results of excitotoxic events, damaging events. When one cell is exceedingly activated by sodium and potassium, that are the two basic ions in the activation of a neuron, and calcium comprised, we have a kind of death-inducing events that are called excitotoxic. That means the excitation is too at high degree for the resistance of the axon, and axon goes under a kind of death that is called, as we all know, apoptosis. Excitotoxic and inflammatory conditions involve an increase in extracellular glutamate levels that produces severe damages on oligodendrocytes, then to myelin produced by oligodendrocytes, as we can observe in the figure, and glutamate is then released again from damaged axons, and we have a kind of a damaging loop generating the definite death of the cell. The excessive astrocyte AMPAR activation, that is in this case not on neurons but on the astrocytes, produces a downregulation of astrocytes themselves. And this means that astrocytes are not more able to have a colloquium, a functional interrelationships with the nodes of Ranvier that are represented by the nude parts between two tunnels of myelin in the A figure. Comparable events characterize the dynamic gray matter. And we can see here the schematic representation of macroglia diversity in gray and white matter areas in the central nervous system, all united in two in two-section figure. One, the left one is for the gray matter, and the other in the right part of the figure is representing the white matter. And this now quite easy to understand the roles played by oligodendrocytes and the astrocytes in the two conditions, as well as the receptor, NMDA, AMPA receptor, caenic acid receptor, CAR, and the gap junctions. Astrocytes are indicated with a yellow border, and oligodendrocyte lineages are indicated with a blue border. As we can see, oligodendrocytes and astrocytes have strict relationships depending on the region where they are, the gray matter or the white matter. Here we meet some embryological note and the topographical imaging of the volumetric schemes of the development of the neural tube and of the telencephalic structures. The figure is extremely complex, and we will afford it step by step. This is the first stage of the series of images that will bring us to the end of the description. As we can see, the figure is divided in two parts, a left part under the name of patterning, and the right part going under the name of neurogenesis and gliogenesis. Let's put our attention to the left part of the figure under the name of patterning. On the top part of the figure is represented the neural tube, that is the precursor of the whole central nervous system, comprised the spinal cord, and then in the apical part of this neural tube will develop the so-called telencephalic structure that is represented in the bottom part of the figure. As first stage, we will go only through the acronyms constellating these two figures, and we will be particularly attentive to their black rectangles, including some explanation. In the top rectangle, we will see that we will consider the so-called BMPs, or bone morphogenetic proteins. That's a group of powerful morphogens, that is substance or elements of chemical elements contributing to the final morphological shaping of the neural tube and also of the telencephalic structures. And these morphogens are critical for the development of the whole nervous system. Then, after the BMPs, we will consider the so-called sonic hedgehogs, that are again proteins, and they are well explained in the second, in the bottom black rectangle, where sonic hedgehog is a kind of protein, or is a series of proteins in humans, encoding by the SHH gene. The acronym SHH is arguably the acronym for the sonic hedgehog proteins that are coded by this gene. This protein plays a key role in the development of the whole animal reign, from insects to mammals. These two kinds of proteins, BMPs and SHH, are extremely important. They are a kind of supervisor for the completion of the shapes and of the region delimitation in the central nervous system. They are in the top part of the list of the proteins, intervening stage by stage in perfectioning the development of the brain, and not only of the brain, but also of the whole body. If we go down into the telencephalic structure image in the bottom part of the left part of the figure, we have the hem proteins that participate to axonal development in the CNS. And these M proteins pertain to the family of the WNT gene family, that is the final black rectangle. The WNT gene family is characteristically expressed at signaling centers that pattern adjacent parts of the neural tube. And we can see that HEM, BMPs and WNTs proteins are particularly involved in the telencephalic vesicles, the blue-red parts of the figure. And there are other factors here, right, like the rostral center FGF8 or FGF7 and TGF-alpha that contribute to the correct development of the brain. And there is also a very important section, the ventral center of SHH, that comes from the floor plate, as we can understand in the upper figure, where we see the floor plate and the violet arrow indicating the transit of the sonic edge of the proteins and their degree of development. Now, let's skip all the other parts, and we will go through these 4 images in detailed way. So let's start with the A figure that shows the primitive neuroepithelium of the neural tube that is patterned by organizing signals that, as we saw before, are composed by BMPs and SHH and by WNT families again. This is just the first image showing the stages of development of the neuronal tube and the particular importance they have in the generating the shaping of the whole central nervous system. Then we go to the B figure. All the figures in details are enclosed within red rectangles, and we see a cross-sectional view of the neural tube that is on the figure on the left of this section. Progenitors of motor neurons and interneurons are formed with distinct regionally restricted domains of the ventral neural tube. In fact, in the ventral part of the neural tube in this section, we can see the different domains of genes and proteins called PZ, P1, P2, and P3 domains that give birth to the interneuron subtypes. And we have also an orange section defined by the PMN domain, that means the P domain for motor neurons. Dorsal domains are also similarly parcelled and are not shown here, just to not make the description too heavy. Signaling mediated by SHH are denoted by purple circles, little spots, as we can see in this figure, in this B figure. On the bottom there is the SHH domain of the genes and proteins on the floor plate of the neural tube, and they little by little go upstairs in some way towards the roof plate, and they dilute in this transit, being very dense near the floor plate and becoming less and less dense in the stages of the transit toward the roof plate. And there is a gradient in some way. And these factors mediated by SHH regulates the expression of transcription factors. For example, we can see here the different transcription factors indicated as the NK X2.2, the OLIG2, the PAX6, and the SCL in the ventral neural tube. And this regulation of transcription factors will be very deeply discussed in further slides. So don't worry to understand immediately these acronyms, because they will become very clear in the next stages of this talk. The interactions of these factors sharpen and maintain the domain boundaries. What does it mean? That we have different regions or clusters of neurons and nuclei that will become mature in the full development of the brain. And what we are interested to here is the development of the glial cells. And we can see here 3 astrocytes subtypes that have been identified. And it is evident that there are different kinds of astrocyte subtypes developing here that are indicated in the pentagonal little figures here, indicated by V1, V2, and V3, where VA astrocytes will express directly the PAX6 factor and reelin derived from P1 that are the most dorsal. Then there are the VA3 astrocytes or astrocytes precursors that express the NKX61 and SLIT1 factors derived from P3. And then again the VA2 astrocytes, which express PAX6, NKX61, reelin, and SLIT1 derived from the P2 type of astrocyte precursors that are located in the intermediate white matter domain. The description appears boring and heavy, but at the end of this chapter we will have all the things made clear, and we will understand why I insist in describing these factors, because they are the crucial part of the developmental stages of the glial cells and of the neurons in the meantime. And finally, we have the embryonic OPCs, oligodendrocyte precursors, that are derived mainly from the PMN domain, illustrated by the oval within the ventral part of the neural tube near the floor plate. And PMN domain is also the orange pentagon, and is recognized by the expression of other factors like PDGFR, A-alpha, SOS10, and NG2. And let's keep in mind that these are crucial factors for the development of glial cells. Then let's go to the C part of the figure, that is the bottom part of the left section of the figure. And we have the organizing centers for the forebrain that will become, in general, the brain, as it is called without the spinal cord. And these include the cortical ham in purple, which is a dorsal source of BMPs and WNTs that we met before. A green ventral center, which is the source, as we saw before, of the SHH proteins, and is also the place of origin of the gene originating SHH substances. And in the rostral, or the pink and blue region, we have the sources of the growth factor, like the FGF8 and the transforming growth factor alpha, TGF alpha, and the FGF7 respectively. Now let's go to the final figure in D, that is representing a coronal view of the embryonic forebrain. The age, the embryonic age of this slice is at 14.5 embryonic day, and its division into dorsal and ventral region shows specialized regions producing different neuron and glial cell types. As we can see, we have a ventral region that includes the lateral medial ganglionic eminences in blue. Then we have a dorsal region that comes before, that is signed in red. And then we have a preoptic area in green, and all these three regions, the dorsal, the medial, and the bottom regions, give rise to different kinds of cells. And we can see them in the big rectangle included in the smaller rectangle. In the red region, that is the dorsal cortical region, is the source of pyramidal neurons and of astrocytes. Then we have the lateral and medial ganglionic eminences, the two bumps within the figure, that give rise to GABA-containing interneurons and, pay attention, to oligodendrocytes. And again, the preoptic area in green gives rise to GABA-containing interneurons again, and again to oligodendrocytes. So in the rostral, in the upper part of the structure, there is the source of astrocytes, and the two bottom parts, the blue and the green parts, have the sources of oligodendrocytes. And even these regions are populated and generated by transcription factors. See later slides for the clarification of these names, that are associated with dorsal NGN1, NGN2, and GLI3, and the ventral patterning substances ASCL1, DLX1, DLX2, and that indicate the fate specification. And there is also a very important factor, that is the NGN, or neurogenin. Now, these were the four very complex and very difficult steps, but we will understand them from these following slides, and we will go through a better definition of the acronyms shown before. So let us ask to ourselves, what do mean the acronyms constellating previous figures? And let's start with a very originating and complex proteins called basic helix, loop helix, BHLH, and other transcription factor proteins. The basic helix, loop helix proteins are dimeric transcription factors found in almost all eukaryotes. Put your attention here. The whole living realm is populated by this special protein, and especially the eukaryotes. They are not contained in prokaryotes. That means that all the cells with a nucleus, a well-defined nucleus, that characterizes the eukaryotes, that means eu stays for the Greek, eu, that means well, and karyon is the nucleus. All the cells with the nucleus, well-defined nucleus, contains these transcription factors that in this case are basic helix, loop helix, defined because of their form. But there is an enormous amount of shapes, and we will see some example here. In animals, they are important regulators of the embryonic development, particularly neurogenesis, myogenesis, herd development, and hematopoiesis. They are characterized, as we said before, by the most wide shape differentiation. Here we have the image, just an example of a myogenesis-inducing BHLH transcription factor. And as we can see in the figure, we have loops defined by these two strings on the left and on the right of the protein, and the remaining parts are helicoidal proteins, interacting often and often with DNA, as it is the case here, indicated in violet. And this is the scheme of what helix loop helix means. Let us put our attention to the A part of the figure, and we can see the helicoidal structure of the protein with its N-terminus, and then the loop part, and again a second helix part with its C-terminus. This is the basic scheme, and over this scheme we have, as we said, an enormous number of structural variations. And let us see the basic helix loop helix family regulating early differentiation, the stages of evolution of neural stem cells. And this scheme indicates the importance in the differentiation of neural stem cells, the NSCs, to glial precursor cells. As we can understand from the scheme, from this cascade, the NSCs, the neural stem cells, undergo different inhibitors and activators, and they become, in case of different configuration and timing of activation of inhibitors and of activators, either glial precursor cells or neural precursor cells. And on the right part of this slide, we can see the olig family regulator motor neurons and oligodendrocyte differentiation. And olig means oligodendrocytes, but it is interesting to note again that this factor that are the oligs are again BH, LH proteins, go under different inducers, coactivators, acting on the neural stem cells. And in this case, as we saw before in the last figure of the last figure that I showed with the 2 slides before, the coactivators of definite types like KX6, 1, PAX6 and NGN2 become coactivators to transform NSCS in motor neurons, while SOX10, OLIG1 and KX2.2 become proactivators or coactivators of oligodendrocytes. So it is very important that the basic helix loop helix family, it must be considered a key role family of substances inducing correct differentiations in the neural stem cells, getting the final stage of glial cells or true neural precursor cells. And as we can see here, we have again a BH-LH protein configuration image that just to maintain it in our mind. And we can see well indicated the loop, but most importantly, let us see how these transcription factors can act in the conduction of the differentiation of precursor cells into mature cells. And we have here the gray line indicating the DNA, and we have a kind of loop and not a straight line as DNA is in usual images. And this is due to the fact that we have a very delicate, the image is indicating a very delicate stage in the stimulation of gene expression to induce the differentiation of cells. And on the extreme left of the figure, we have a DNA binding protein that is a special protein that attaches in some way to the DNA and provokes the bending of the DNA itself, and the DNA is now a loop. And there are many oval and round molecules getting into this kind of space that is composed by transcription factors that are proteins that control the rate of transcription from DNA to messenger RNA by binding to specific DNA sequences, the so-called activator binding sites, or the block of these binding sites called globally the enhancers. The function of these transcription factors is to regulate, turning off and on, the genes in order to modulate, increase or reduce their expression in the right cells at the right time in the right amount. Here on the left is represented in a simplified way the machinery of a transcription factor. As it is highlighted in the next slide, the transcription factor machinery could be much more complex than shown in this scheme. And again, staying on this figure, we can see in the bottom part of this DNA loop the promoter region, the gene region, and an RNA polymerase. And all these factors, the transcription factor, the mediator proteins, the DNA promoter regions, and the RNA polymerase interact among themselves, and we cannot go in depth, otherwise the lesson could be much longer and out of our goals. We come to the stimulating transcription of the gene, and transcribe the gene going into mRNA give rise to the right proteins, inducing the differentiation of cells. Let us see in another image, just to fix in our mind the transcription factors of eukaryotic cells. We can see the 5 steps that are involved in this looping of the DNA. Note that this diagram simplifies the DNA greatly, as it simplifies also promoters, enhancers, insulators that can be dozens or even hundreds of base pair long. That means they can occupy longer tracts of DNA. And again we have in 1 on the top of the figure the activator proteins binding to pieces of DNA called enhancers. Their binding causes the DNA to bend, bringing them near a gene promoter, even though they may be thousands of base pairs away. And then we have other transcription factor proteins in 2, joining the activator proteins, forming a protein complex which bind the gene promoter. In 3, we have this protein complex that makes it easier for the RNA polymerase, the orange or rose-orange blob, to attach to the promoter region and start the transcription of gene. In the 4th part, we have an insulator part of the DNA with methylation group, and these can stop the enhancers that are the starter of the gene transcription, and blocks and can stop from binding these enhancers to the promoters. And if a protein called CTCF, named for the sequence of the parts of the bricks of the DNA, CCTC, which occurs in all insulators, binds to it. And we have CCCTC means obviously cytosine, 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 thymine, cytosine. Methylation is the final stage, that is the addition of a methyl group to the C nucleotides that prevents the protein called CTCF from attaching to the isolator, turning it off, and allowing the enhancers to bind to the promoter. And this is the kind of opposite stage of the fourth part of this figure. This diagram, as we said, simplifies. And let's now face another issue involved in the evolution and development of glial and also of neuronal cells. The many external factors that regulate the glial development play, in fact, different roles in the different stages of maturation during the cortical and the subcortical development. We will see here a global sector dedicated to delicate arguments of the activation and inactivation timings of developmental genes and proteins in the maturation stages of glial cells, and necessarily also of the neuronal counterparts. What I'm going to show you here is just a mere example. Here is a single developing factor that has been chosen, and just as a pure example, not because a particularly prominent role. The so-called lethal giant larvae 1, LGL1 factor, induced during oligodendrocyte differentiation. The strange name is due to the fact that it is so-called because of its first detection in Drosophila melanogaster larval development cells. And this introduces us to a kind of denomination problem of molecular biology. This example is, in fact, used to show how the denomination of a factor, a maker, or a promoter, or else, takes the name from the circumstances, biological species or organ or else, in its first detection, but then showing profoundly different biological assignments in different contexts, where it is then observed in different researches and in different contexts, where is then detected or observed. And the LGL1 function is required for ventral zone integrity, that is the controlling zone of embryonic neurogenesis. LGL1 regulates, in fact, cortical astrocyte production and controls postnatal neurogenesis in ventral and subventral zone, ventricular zone. And you can see in the figure the acronym LGL1 in black, and its main roles that change following the embryonic development. And the embryonic development is indicated by the letter E8 to E10, E12, then 14, 17, and with birth, and the postnatal stages in adult progenitor. There are discrete and distinct sequential functions of a glial development controlling factor LGL1 in regulating radial glia behavior in the development of neocortex at distinct stages of cortex development. And we can see that LGL1 can give birth to neuroblasts and climbing neurons in the second image of the figure, where we see the astrocytes, the radial astrocyte, acting as a scaffold for the blue neuron or neuroblast climbing towards the cortex. And then again, there are other stages where the neurons are getting little by little the superficial layers of the cortex and the cortical plate. And then again, in the fourth stage, E17 and birth, we can see that LGL1 favors not only the climbing neurons along the radial glia, but also the generation of ramified astrocytes. And postnatally, LGL1 favors and promotes the generation of neuroblasts that will contribute to the final stages of the cortical development. Let's add another new interesting feature, that macroglia in the gray and white matter display regional plasticity and intrinsic heterogeneity. What do they mean, these two terms? Plasticity indicates the adaptation of the same cell type to the local functional needs and regulating also the responses to injury, while heterogeneity means and indicates the intrinsic regional transcriptional differences in cell populations with consequences for cell functioning upon CNS injury, such as demyelinization or remyelinization, or demyelination, remyelination, as are also indicated. For instance, in leukocortical multiple sclerosis, that is, multiple sclerosing involving mainly the white matter, lesion in the cortex, in the GM or gray matter area, remyelination is more efficient than in the white matter, as in the corpus callosum, the white matter area. Let's step now into a deeper stage of glial embryological and adult functional details for glial shaping and dynamics. We shall afford now just specifically the features of oligodendrocytes and their precursors, and then of astrocytes. This shows the background to the manifold roles of glial cells. Let's start with oligodendrocyte lineages with embryological and developmental features. There is a number of fundamental roles of oligodendrocytes that are well known, but we are very far from knowing all of them. And currently we know the intervention or the many roles played by this kind of glial cells, from the axon myelin sheathing to synapse modulation and neurotransmission in both gray and white matter. Specific markers help at either identifying or classifying them. In this summary, I will propose the main types of markers, biochemical markers, characterizing the progressive oligodendroglial lineage maturation. And then we will see in details these acronyms that have been already shown before in a more general presentation. So the transcription factors OLIG2 and SOX10, and you have to wait for the next slides for this marker short descriptions. Then we have mature oligodendrocytes developing from their precursors, which are supported by PDGFR alpha and NG2, that is also known as CSPG4 expressing cells. Throughout all the maturation processes, these cells pass an immature pre-myelinating stage identified by the transient expression of another factor, BCase1, or breast carcinoma amplified sequence 1 protein, also called, put very well your attention to these words, to brain-enriched myelin-associated protein 1, encoded in human by the BCase1 gene and by the ENPP6 gene. At this intermediate pre-myelinating stage, the myelin lipids sulfatide and galactosyl ceramide are already present at the cell surface. Myelinating oligodendrocytes are recognized by their expression of myelin-specific protein. That is most important, and I ask you to keep them in mind. The membrane basic protein and the proteolipid protein that have the obvious acronym of MBP and PLP. Let's understand what OLIG2 means, this factor, this crucial factor in the oligodendrocyte development. Oligodendrocyte transcription factor 2, this is the meaning of the acronym, is a basic helix-loop helix transcription factor protein. I have not to explain again this complex definition because we faced in the slides before these terms, and these are encoded, this factor, transcription factor protein is encoded by the OLIG2 genes. There are other OLIG genes pertaining to the BHLH subfamily that are OLIG1 and 3, and we will not consider them. The expression of OLIG2 is mostly restricted in the central nervous system, where it acts in some way as both an anti-neurogenic and a pro-neurogenic factor. This is an apparent contradiction, but it is not a contradiction because these two roles, these two opposite roles, are played at different stages of development. We noted just before how the different stage of development can change the role of a developmental factor. And in this case, OLIG2 determines motor neuron and oligodendrocyte differentiation. You will remember in the previous figure how motor neuron and oligodendrocyte differentiation come from the top parts of the developing brain, the cortical part of the developing brain, and where motor neurons and oligodendrocyte differentiate deeply and in fast way from the same pool of progenitors. And the OLIG2 factors has also the role in sustaining replication in early development. I put here on your right in the figure again the image of the BHLH schematic image and a schematic image of a transcription factor as a whole. We have already seen these figures and we now know what they mean. In pathological conditions, OLIG2 may fail in producing these two opposite effects and becomes involved in diseases such as brain tumors and Down syndrome. Apart from functioning as a neurogenic factor in specification and differentiation of motor neurons and oligodendrocytes, OLIG2 factor also functions as an anti-neurogenic factor, as we before noted, at early time points in the precursors of motor neuron, the progenitors of motor neuron, to sustain the cycling progenitor pool. This anti-neurogenicity of OLIG2 plays an important role also in tumors and especially in gliomas. This is again an indication of details of the BHLH transcription factor and an overall transcription factor scheme. Let's understand what OLIG2 means, this factor, this crucial factor in the oligodendrocyte development. Oligodendrocyte transcription factor 2, this is the meaning of the acronym, is a basic helix-loop helix transcription factor protein. I have not to explain again this complex definition because we faced in the slides before these terms. And these are encoded, this transcription factor protein is encoded by the OLIG2 genes. There are other OLIG genes pertaining to the BHLH subfamily that are OLIG1 and 3, and we will not consider them. The expression of OLIG2 is mostly restricted in the central nervous system, where it acts in some way as both an anti-neurogenic and a pro-neurogenic factor. This is an apparent contradiction, but it is not a contradiction because these two roles, these two opposite roles, are played at different stages of development. We noted just before how the different stage of development can change the role of a developmental factor. And in this case, OLIG2 determines motor neuron and oligodendrocyte differentiation. You will remember in the previous figure how motor neuron and oligodendrocyte differentiation come from the top parts of the developing brain, the cortical part of the developing brain, and where motor neurons and oligodendrocyte differentiate deeply and in fast way from the same pool of progenitors. And the OLIG2 factors has also the role in sustaining replication in early development. I put here on your right in the figure again the image of the BHLH schematic image and a schematic image of a transcription factor as a whole. We have already seen these figures and we now know what they mean. In pathological conditions, OLIG2 may fail in producing these two opposite effects and becomes involved in diseases such as brain tumors and Down syndrome. Apart from functioning as a neurogenic factor in specification and differentiation of motor neurons and oligodendrocytes, OLIG2 factor also functions as an anti-neurogenic factor, as we before noted, at early time points in the precursors of motor neuron, the progenitors of motor neuron, to sustain the cycling progenitor pool. This anti-neurogenicity of OLIG2 plays an important role also in tumors and especially in gliomas. This is again an indication of details of the BHLH transcription factor and an overall transcription factor scheme. Then we have SOX10 that we have already met before without any explanation before and now getting into its explanation. SOX genes are the acronym, there is the acronym of SOX meaning SRY related HMG box gene. HMG means high mobility group box. There are genes that can move within the context of DNA, and this is a protein domain which is involved in the DNA binding, encoding a family of new transcription factors that bind to a minor groove in DNA. In DNA there is a major groove that gets the messenger RNA and the minor groove that has the important role to be regulated by transcription factors. The protein has a rich array of expression and modulation domains. Now let's come to understand what SRY means. SRY means sex-determining region gene, and I illustrated here the gene, the Y gene, the sex Y gene, and the region in blue of the sex-determining region Y. And it is called like that because it was first discovered in this chromosomal region. But as we can understand easily, this is not devoted exclusively to functions related to sex-determining Y chromosome. And we have on the figure on your right the a representation of the steps and the importance of SOX10 roles. We have on the very top of this figure the neural crest cell, a kind of omnipotent progenitor that can give rise to glial cells and to neurons. And as we can see, the protein SOX10 can give rise to several stages of development with different ending points. At the extreme left of the figure, there is the first developing cascade of SOX10 that acting on other developing factors is expressed in migrating neural crest cells and persisting until the full development of neurons and of melanocytes that are melanin synthesizing cells in these red rectangles. Then we have the stage of maturation of gliogenesis in peripheral nervous cells, PNS indicated here. In the peripheral nervous system, myelin, it regulates the expression of the Schwann cell-specific myelin genes, PZ, myelin basic protein, and proteolipid proteins, all terms that we have already met before. This is why I insisted before in presenting them to you, even not explaining them diffusely, but just to indicate their crucial roles, the crucial interventions in the developing of the different parts of the central and also of the peripheral nervous system, as here in this new rectangle. And as we can see, this stage of red, the second red rectangles brings to gliogenesis and to the maturation of the peripheral Schwann cells that are the glial cells of the peripheral nerves. SOX10 is also involved in the autonomic nervous system, where it regulates the expression of MESH1 and FOX2B transcription factors necessary for sympathetic nerve fate. NS cells, as you can see in the violet blob under the blue rectangle, means in fact autonomic nervous system. And as we can see, there are many parts that are involved with the intervention of SOX10, getting to the formation of sympathetic nerve fate, the enteric nervous system development, ENS, and the GDNF signaling that we will consider just after these stages. SOX10 is, as we have seen, involved in the autonomic nervous system, when it regulates, as we have seen, MESH1 and FOX2B transcriptor factor. In the enteric nervous system, SOX10 interacts with PAX3 to activate the GDNF-CRET pathway. It also regulates endothelin receptor beta during the enteric nervous system development. Arrows indicate positive regulation by SOX10. And then there are a lot of other acronyms that involve the development of these autonomic nervous system sections. SOX10 is expressed during the specification of migrating neural crest cells. As we have seen before in the top part of the figure, there is then a symbolic drawing of the neural crest cell, and the neural crest cell is a kind of progenitor of both neurons and glial cells, and they migrate along the neural crest in the embryonic early stages of development. And it persists in neurons and in melanocytes, as we have seen. In this slide, only 2 points must be enlightened. And let us put our attention to the section A in the figure on the left, where the cranial neural crest domains, that is, the various stages of the migrating neural crest cells, appear as 2 stripes, violet stripes in the figure. And these are pictures, histological pictures taken from the studies on developing zebrafish embryos. And then we have the section D we must put attention at, where there is a lateral view of the zebrafish head depicting the same migratory path illustrated in A by the violet stripes that develop in their migration routes to form the cranial neural crest in purple, and we can see them as spots, violet spots that go from the orange region toward the back part of the sagittal, of this sagittal